We've gotten a number of questions from the webcast, and we're very pleased with that. And so I'm going to try to intersperse a few of those. If there are people who are in the audience that want to ask questions, come up to the microphone. We've got about 10 to 15 minutes for questions, so I'll ask you to keep the questions relatively short and the answers relatively short. So let me just start off with one that came in from Ann Summers at University of Georgia. This is for Dr. Knight. Are registrants' filings checked regularly or randomly, especially the non-standard results outcomes, by any independent organization?
Okay, well, to answer that, ECHA have a systematic program where we have to do what's called a compliance check on a set proportion of the dossiers. In fact, we're supposed to do 1 in 20, which is 5% in each tonnage band, okay? Can you hear? Okay, nod. Yes? Okay, you can hear, good. We select those partly at random to keep industry on their toes, as it were, and the other is targeted based on criteria that's in the legislation. For example, if they're on the CORAP list of substances that are going to be evaluated. And the other one is we target them based on concern-driven criteria. And there's going to be a new system where we target specific endpoints, specific areas, doing a targeted approach. So the question, so that's how they're targeted. The question is, does somebody else independently check them? No. It's our job to check them, but all the information is disseminated and published on our website, and we know that certain organizations have checked samples of them and in fact published their own assessment of whether the read across and categories data is okay or not. So I hope that answers the question.
Thanks, Derek. The second part of the question from Anne was briefly, how are inorganic toxics such as metals and halides handled in the REACH system, particularly with regard to the read across groupings? Okay.
Well, basically inorganic substances, metals, metallic substances, and organic substances, in a sense, legally are all treated the same. Administratively, they're all treated the same. And there's actually often an issue of substance sameness and when does a substance become a different substance. But I do understand the concern, because sometimes the traditional toxicological assessment, the traditional testing is more focused towards organic substances. Anyway, to cut a long story short, the inorganic substances, the metals, metal salts, etc., in theory, a registrant would have to provide the standard data set, but if they can establish the properties, the environmental or the human health properties can be assessed by categories or read across, then they are at liberty to do that. The challenge actually is a very practical challenge, isn't it? Because if you've got metal substances, the solubility can dramatically affect the bioavailability for human health and the environment. And I know there's quite a lot of work being done. I think they're called HERAG and MERAG, which are designed to model the effect, the dissolution effect of metals in simulated physiological fluids and for the environment. And that can serve as a model for working out the actual exposure. So the short answer is, yes, you treat them in the same way, but the actual scientific techniques that you use for categories and read across will be different. But it's up to the registrant to make the scientific case.
So let me just bring in one more question from the web that sort of follows on from this, and it's really directed, I think, both to Derek and to Isla. And this comes from Cindy Barnes at Health Canada. The gist of the question is, doesn't use of groups of chemicals or representative chemicals to predict a hazard using read-across type of categories suggest that we really ought to be looking at the implications for cumulative exposure among those groups as well?
I would say yes.
Okay, that's a short answer. Derek, do you have a thought about that one?
Yes, the thing about REACH is the responsibility for establishing safe use rests with the individual registrant. So if there's 20 companies all supplying the same substance, what they have to do is make sure that the risk for their use, manufacturing use, is okay, is adequately controlled. If the regulators, by that I mean the member states or the European Commission, think that there's a possibility of a problem due to cumulative effects, they can perhaps put the substance in for substance evaluation, where those ambiguity in the risk can be taken into account, and one of the elements could be cumulative risk, cumulative hazardous properties, cumulative effects. And indeed, if that's established to be the case, or there's other evidence already, it's already established that there's a risk from cumulative effects, then the other risk management processes, the marketing, the restrictions process, or possibly even the substances of a very high concern process, but usually the restrictions process can be invoked. And the European Commission are actually doing work at the moment to assess whether EU legislation is okay in terms of dealing with these these cocktail effects. But in terms of the opportunities already within REACH, there are regulatory instruments that can take this into account.
So let's move to the floor here. Stan?
Those kinds of efforts as you move forward. This is more of a process question.
Derek, did you hear that question?
No, I got absolutely none of it. I'm sorry. Could somebody repeat it?
So just a quick repeat and then perhaps a quick answer. Stan Barone's question was, how is CBI, confidential business information, treated within REACH, a process question, given the way that the data is made available?
Okay, well, you can check on our website and see chapter and verse on it, all the details, but basically a company can claim certain information as commercially confidential, such as detailed use information, and under very certain specific circumstances they can even claim the chemical structure as confidential. And then when they submit the registration dossier, they submit that confidentiality claim and they have to justify it and pay a fee. And then we assess the claim. In the meanwhile, the parts of the dossier that are claimed confidential are not disseminated. If the claim is rejected, then the dossier is updated and that information would be disseminated. And there are sort of appeals processes and everything that can be done with that. But basically the results of the studies are disseminated, including the robust study summaries unless a company claims that as confidential, but the commercial information on the detailed use pattern, etc., or in possibly the impurity profile or whatever, can be claimed as secret.
I guess, Derek, what I'm trying to get at is that causing any kind of impairment in the read-across efforts and in the evaluation of the alternative tests that CBI claims? That's what I'm trying to get at, not the CBI rules that are posted on the website. In your
well, the short
experience
answer,
thus far.
Yeah, in my view, it hasn't caused any problem because virtually all the information is disseminated, and there's only a very, very small number of substances where the chemical name is claimed as confidential and not disclosed. So basically all the information is there. An area that the potential problem is that the quality of the information that's disseminated, in other words, the level of detail and so on, is a little bit variable. And one of our themes is to encourage companies to update their dossiers and improve the quality of the registration dossiers. Because basically the information that's disseminated is just lifted straight from their dossier and then disseminated. So normally, well, we don't actually change or intervene to correct errors in the dossier. Obviously, if when we actually examine a dossier and there's a problem with it, it may be that the company would then, we would ask the company to update and correct any errors that have been identified that are subject to a quality observation letter, or update the dossier with new information. But there's no systematic check by a third party of the correctness of the information put into the dossiers.
A quick question to Isla. Isla, in your conceptual model, you had identified value of information as an approach being incorporated into the evaluation of the traditional, non-traditional efforts, particularly, I think, in Tier 3, but informing the other tiers. You didn't really talk about that in your talk, and I was wondering, is that transdisciplinary approach included? Is that just a future effort, or what?
Well, I would say that we probably have not done as much as we need to do, but I think understanding which information is valuable, quite literally, among all the information that's available, is going to be key in terms of moving forward at all three tiers.
And we'll be talking about that with a specific talk and a panel discussion tomorrow,
right.
So that's going to be important for us. Richard?
Hi, Richard Dennison, Environmental Defense Fund. This is a comment really probably for Isla. Is that going to go away? Let me try to talk over it. Isla, it has to do with your depiction of the tiers and the statement that greater uncertainty is okay more on the screen, the Tier 1 and then on the other. And while I think as a general proposition that's probably accurate, it seems to me there are some policy calls almost that underlie that. So for example, you mentioned that under ToxCast currently the false positive rate is much better than the false negative rate. That is a little troubling to me in the sense that if you apply a screen where you know everything that gets through that screen and moves on is going to be evaluated further, you really want to make sure you have a very low false negative rate. It sort of picks up on something Kim said earlier that the industry fear about all this is that any little blip is going to flag a chemical. On the other hand, if you're using it for a screening purpose, maybe you want that sort of thing to happen, and then you weed those out later. So I guess my question is really, how is the agency thinking about a set of decision rules and really what come down to a set of policy decisions about the relative amount of uncertainty and the nature of that uncertainty, both communicating that and also deciding sort of what's acceptable and what's not?
Now, I think unfortunately effectively it's going to be defined in practice as we get some specific examples and uses in different situations. Some of that will become clearer. But I have to say the issue that you raised about screening large numbers of chemicals, and it's nice that you have a great deal of confidence that when there's a positive finding that chemical is disrupting an important biologic process. But I think one of the things that's kind of disturbing all of us is what about the chemicals that you miss? And the only thing I can say there is that we're continuing to proceed to improve the screening methods so that we can, I think in the near future, we'll probably have to go back and re-look at chemicals as knowledge develops.
So, Eiley, we had a similar question from Amy Kyle at Berkeley, and I think you've answered that with the response to Richard. So, Joyce?
Joyce Suji, Exponent. I wanted to thank the speakers for their very interesting and thought-provoking talks. In reflecting on it, I really enjoyed hearing about the attempts to look at more environmentally realistic exposures and what's going on at the cellular molecular level and what are the pathways that are happening at these exposures. But I think it also underscores that we need to do better exposure assessments at the cell and molecular level and not just take drinking water concentrations and expose that to cells, which is what is going on right now. And often that is not the concentrations that the cells are actually seeing. So I think there's a lot of challenges and a lot of great research going on a number of different fronts that will come together and hopefully inform us better so we can make these risk assessments more accurate. My second comment was I think the three chemicals, Isla, you mentioned, benzene, ozone, and pH is the first two. We do have a lot of good human data, which is great. The third one, I hope we're not trying to validate the new paradigm with old paradigm data based on high-dose gavage corn oil and forced stomach tuners and rodents. That's my one comment about that. So I think with the other two, I think we're safe. We have a lot of good data at a number of different doses in humans, and we have more information. On PAHs, I'm less sure.
And Joyce, that same question about the source and availability of exposure surrogates was raised by Mike DeLarco from the National Children's Study on the web, and so that was a question he wanted to get a comment on. I think it is a legitimate concern as we begin to think about these kinds of issues.
Mostly because of time, I didn't go into exposure research also, but people are certainly aware of that as an important issue and doing a lot of work in that area. The second comment on the PAHs, as much as we've studied PAHs, we were a bit dismayed about the data that were available and wished there was more and better data. But, it was really an attempt to look at various PAHs to see if you could see a molecular pattern that you could then begin to explore the attributes of different kinds of mixtures. But I would say that project is still the furthest behind.
So we're going to do one more from the web and then take our final question from the floor here. Lauren and Derek, Jennifer Shee from Cal EPA asked about some practical examples or potential methodologies to address the issue of population variability and general background exposures using these kind of systematic approaches that are being discussed this morning. Any comments on that, Lauren?
Well, I think we're going to hear through the day, particularly maybe with Dean Jones' talk, a way into thinking about background. And we did have an Exposome conference that I think would be useful to look at and consider sort of indicators of the background status. In addition, there are some cell systems from which genetically diverse cell systems, and Yvonne Rusin has been involved in looking across, and there are cell systems from Europe and from the U.S. That you can look at genetically diverse responses in these cells. So there are publications available to look at.
I think those are going to be some systems that will be valuable to address some of those issues.
Hi, Jerry Poggi, SOEH. And maybe Derek, I'd welcome your thoughts about how are we going to get to a different side of the equation. That is, Eric Schott has recently looked at a fair amount of information about pharmaceutical developments, looking in a similar way at pathways that are putative therapies, but looking across multiple possible therapeutic agents to optimize for that will do the best good and cause the least harm. And I guess my question is, are we going to be simultaneously developing algorithms that would allow for choices to be pursuing least toxic alternatives as opposed to defining to the nth degree the hazard of a single agent? And how do we gain the advantage and perhaps the competitive advantage for the broadest societal good for developing new products with least toxicity quotients?
That's a really good question. For those of you who are perhaps not familiar with the Chemical Safety and Sustainability Program, which is a larger effort within EPA, many of these tools are targeted to the kinds of questions that Jerry's talking about, where you can look at essentially the risks and benefits of various chemicals, and perhaps in advance of putting chemicals out in the market, make much smarter decisions about which chemicals you want to use. So, I mean, that's an important focus. But I think the other thing that you raised is the research that's going on in the pharmaceutical industry that's looking at multiple chemicals. And I think our problem in environmental toxicology is exactly the same, and that those same approaches should be adapted. And, we plan to adapt in terms of looking at mixtures, and mixtures could be chemicals that on the surface look completely unrelated but disrupt the same biologic systems. So I think there's a long process there, but I think it is a path forward.
So I think we're going to have to end up this morning's session. I did want to let Kim know that there had been a comment and question raised shortly after your presentation. It was from Ann Summers at University of Georgia. and it related to the role of the microbiota in terms of processing toxic chemicals and how that fits with the kind of discussion that we're having right now. And I'll just say to Anne and to others who submit comments, these are going to be comments that will be fed into the process and follow-up discussion from this workshop. So we won't take an answer to that right now, but it is noted as an important issue and one of the issues that we took up in a previous workshop with this group.